Good evening, and big thank you to both Torrent and Dr. GVK. So I'm talking about a topic which is a little bit off the beaten track. Now that semaglutide has gone off patent, we've been hearing a lot about solid phase protein synthesis. I think it's time we sort of revisited our pharmacology roots a little bit, considering we'll be dealing, and we have been dealing with a lot of these proteins and peptide molecules over the past few years. So I'll be talking about that. This is my disclaimer. Of course, this is a program that is sponsored by Torrent, but I've put in some slides to better my understanding and hopefully your understanding about protein synthesis. So what do we mean when we say peptide? Peptide is different from, if you take a molecule like metformin or aspirin, it is what is called a small molecule. So there the weight is around 150 Daltons. If you take a peptide, it is somewhere between 500 to 5,000 Daltons. So for example, semaglutide is around 1,500, 1,800, something like that. And then you have the huge molecules, the monoclonal antibodies that are used in oncology, rheumatology. Those are around 150,000 Daltons. Those are the larger protein molecules. So these are what are called peptides. And a lot of the peptides are hormones, for example, oxytocin, vasopressin. So these are the typical peptides that we think about, apart from, of course, modified GLP-1 and GIP and so on. When you talk about the early era, we've been using peptides for the last almost 100 years. Insulin is probably one of the earliest peptides that we have been using. And, but apart from that, they used to get other hormones. For example, they used to get ACTH from pituitary glands. They used to get calcitonin. Even to this day, we use calcitonin that is removed from the salmon thyroid. So we still continue to use hormones that are extracted from organs. And it's interesting when you think about it. If you just take, like for example, if I give you a pancreas, how are you going to isolate the insulin from it? And this is sort of one of the analogies. Imagine that you have a mixed bag which contains sand, iron fillings, plastic beads, salt. So you have to use different things to remove each one of these organs. For example, you use a magnet, obviously, to remove the iron filings. You'd use water to dissolve the salt. Similarly, you have to use multiple chemical methods to actually extract the peptide of interest. So for example, if you talk about insulin, which I thought is interesting since a lot of us will be using insulin, the first step that is done is something called an acid extraction. At that very low pH, what gets destroyed is enzymes and many proteins, but interestingly not peptides. The fact that it's very small actually protects it from being destroyed or deconstructed in acid. The next step is something called an alcohol fractionation step. At this step, the lipids get precipitated. Again, small peptides remain stable. The third step is removing it. You remove each protein according to the isoelectric charge. Now, what I found fascinating about this was at each step, whatever remnant you get, you have to bioassay it. In those days, they would literally take those bioassay, whatever the remnants they got, they would inject it into lab animals to see if the blood sugars dropped. If the blood sugars dropped to that extract, it contained the peptide of interest, in this case, obviously, insulin. And then you would do. So it's a very cumbersome process. Some of the people here may have used animal insulins, the insulin that was derived from pigs and cows and so on. The big shift, the first big shift that happened with peptide synthesis was when people got to know about the structure. So for an example, one of the first peptides that was synthesized was oxytocin. Oxytocin and vasopressin are very short chain amino acids. Vasopressin, for example, only has 9 amino acids. Oxytocin also very similar. It's got a very short chain. So you could easily add. It's sort of like the analogy that I got was, when people are stringing flowers, I don't know how many of you know how to, we string the flowers. You keep a flower by flower and you make that little like garland or something. So like that, you just literally added amino acid by amino acid and you could make these short peptide chains. The problem was the yield. Every time you started off, let's say you started off with some 100 initial starting amino acids, by the time you got to the end, you would only have, for example, 12 peptides of the final molecules, because at each step, the things would get washed away. So the yield was very low and it was very cumbersome. At each step, you had to add something which protected the amino acid on one side, so that the coming in amino acid would only act on the proper side. Then you have to remove this protection agent. Then you have to modify the second amino acid so that only this protein, the next amino acid would come and get attached. So it's a very cumbersome step-by-step process. The other thing about the native peptides, and the next big shift that occurred was they wanted to see how they could modify the peptides. We know that peptides have a very short half-life. Insulin has a half-life of a couple of minutes, the GLP-1 again a couple of minutes. Most of the peptides which work on the cell surface, they just come, it's literally like an electric charge or a flash of light. They come, they bind to the receptor, and the downstream signaling pathway is set in motion, and then the peptide gets destroyed. So the next step, they wanted to see, apart from the synthesis of the native peptides, they wanted to see if they could make a change in the way the peptides were working. And this is where the role of peptide design started coming in pharmacology. So you wanted a peptide that was more stable, that wouldn't get degraded, that would have a long half-life. So you would substitute amino acids, you would add linker molecules. You are very familiar with this with the insulin analogs. That is all a designer peptide, the insulin analogs. Another interesting one I thought I'll just share was another hormone, a modification of our native vasopressin, where the L-arginine is substituted with a D-arginine. Again, one interesting fact, I'm sure some of you may know about it, most of amino acids in nature, I think literally apart from 3, like D-ribose or something, most amino acids in nature exist in a LEVO configuration. That is where the groups are sort of rotated in such a way that's called a LEVO rotation. Very few are in the dextro rotation. But anyway, coming back to desmopressin, so this had a longer half-life, and you removed one amino group so that the desmopressin acts preferentially only on the V2 receptors in the kidney versus acting on the V1 receptor in the vasculature. So you would not get a vasoconstriction. If you use pure vasopressin as is used in the ICU, you get vasoconstriction. If you use desmopressin, you get the effect that you would want to get for diabetes insipidus. So this was one of the earliest examples of a peptide modification. Of course, there are many other modifications. This is a busy slide. If you just focus your attention on the last column, I've just put some of the peptides that we are very familiar with. For example, insulin analogs, pegvisomant, modified albiglutide. So they all have modifications of the native peptide, which have, like I said, either increase the half-life or reduce the destruction by the native enzymes. And of course, there are other modifications that are done, for example, with tirzepatide. I'm just taking another example where you improve the receptor specificity. So you design the peptide in such a way that it is able to go and attach to its own receptor without having any crossover effect. Now that you had a way to design the peptide of interest, you can modify it in such a way, we come to the synthesis part of it. This is also an interesting journey, I would say. In earlier days, apparently all of it was done literally in beakers using fluids, and that was a very cumbersome, what I told you earlier, type of a process. Till about these two, what is called a solid phase, I'll talk about it in some detail, and the recombinant DNA production. Recombinant DNA really took off because of insulin, and this started happening, I believe, from the mid-80s. The recombinant basically uses a E. coli or the yeast into which you put the vector which contains the gene of interest, so that when the cell starts multiplying, it makes the exact same molecule for which you have added the DNA into the gene. The big advance that came was something called a solid phase chemical synthesis. Again, the person who invented this got a Nobel Prize in 1984. And in this, very simple, if you think about it, the first amino acid just gets mobilized onto a resin bead. That's all. It gets mobilized, it doesn't get washed away. So literally, it's as the name suggests, it's a solid phase chemical synthesis of the peptide, or solid phase synthetic synthesis. The advantage is you don't need to keep removing the protein, you don't get washed away, so you have more of the growing peptide retained. The waste products gets washed away. Obviously, I'm very simplifying it, but this simple modification meant that we now had a way where we could manufacture larger numbers of proteins. This is just a column that I put up talking about where we can use each. The early one was that liquid phase chemical synthesis, which was a very slow process. If you look at sort of the fourth row, you can see speed is very slow. The solid phase, it's faster. If you look at the recombinant DNA production, it's extremely fast. The main advantage of the recombinant DNA process, which we still use for insulin, is that once you set up that initial phase, it is very easy to magnify. It's very easy to multiply. The solid phase peptide synthesis, the initial cost to set it up is low, but you're restricted by the amount of peptides that you can add. So you cannot make it up to 50 amino acid peptide chain is very good with the SPSS method. Anything more than that, it is better to go to a recombinant DNA methodology. Semaglutide, I believe, is what, like 31 or something amino acids. So this is the reason why the solid phase peptide synthesis is really taken off, and most of the peptides we are going to use nowadays, there's nothing called a synthetic peptide. They are either manufactured by the recombinant DNA or they are made mostly by the solid phase peptide synthesis mechanism. And again, in the last row in this, if you look, I've put solid phase synthesis is used for example, vasopressin, desmopressin, all of the GLP analogs, particularly semaglutide, but even the older ones, the liraglutide, all of that. Recombinant DNA is still used for insulin and monoclonal antibodies because those are more complex molecules, and the biological organism has a better way of making it without any mistakes. So medications like semaglutide and tirzepatide rely entirely on the synthetic precision of this SPSS, where you have some amino acid substitution, you have a place where a linker molecule has to get attached so that the fatty acid chain is attached. All of this is possible by first building that backbone using the SPSS method and then adding the side chains. Recombinant DNA, similar thing happens, except that the backbone is made by the yeast, then you purify it, and then all of these modifications are done in the chemical way. So it's really a semi-synthetic is what I would say. So I think the question that all of us were asking when these the semaglutide generics have entered the market is, does the origin pathway matter? Whether it's fully synthetic or semi-synthetic, does it alter the clinical nature of the drug? Ultimately, what decides whether it's going to work well is the final molecular structure, and the final molecular structure is identical between the two. There are a lot of detailed testing that is done, including, for example, mass spectrometry. Now, mass spectrometry is very, very good for picking up even tiny little differences between two different peptides, even a little bit of an amino acid which is out of phase or something. The mass spec will be able to pick it up very clearly, and all of the companies which are manufacturing it have to go through the rigorous standards to prove that their product is equal to the originator product. So, for example, like I said, spatial structure, purity levels. You add a lot of chemicals, you have to keep washing it off, making sure that it is not retained in the final product. But ultimately, if it is I've given the same analogy that two keys which are cut in different workshops but are of the same shape will ultimately open the same lock. And also efficacy is determined not only by all of those physico-chemical methods like mass spec, but also biological methods. So they do like cell-based assays, and of course there are the real-world assays. And if the structural profile and the binding affinity match the innovator molecule, we know that we have something which is going to act very similar to the original molecule. Safety and immunogenicity, this is also important for particularly the peptides which are synthesized using recombinant DNA. Even in this, they've done the studies. The company manufacturing generic have done the studies. It is important we make sure that it doesn't contain some impurity which may not have been picked up by the mass spec or of all of the other methods, but which is causing our body to make some antibodies. So all this has also been tested with the generics. So coming to the clinical impact of the GLP-1 receptor analogs, all of you must be aware, I'm sure all of you are using it. We know how it works. It basically reduces appetite, slows down the digestion, makes the body make more insulin. We know about all that. The GLP-1 analogs, we've been using it for 15 years. And it started off with exenatide, which we had to take twice daily. Then we came to the liraglutide, the lixisenatide, and then the dulaglutide, and now we have the semaglutide and the tirzepatide. These are designed in such a way that it improves the half-life so that we can give it once a week. And they are more potent because of the way they work on the GLP-1 receptor. So semaglutide is believed to represent the peak of GLP-1 engineering, and again, like I said, I think it is achieving remarkable results, not just in diabetes management, but of course multiple other things, as you may have heard in many talks, and it does have quite a long half-life. Of course, the companies need to make sure that the manufacturing can be sustained, The next few slides are about the specific molecule which is being manufactured by Torrent, which is the Semalex, which is one of the brands that they are marketing of semaglutide. This is the way the pens are available, a 2 mg pen, a 4 mg pen, and an 8 mg pen. The 2 mg pen has the dosing for 0.25 and 0.5. For those of you who may not have seen it, the 4 mg pen contains 4 doses, and each dose will give you 1 mg. 8 mg pen, again 4 doses. Each dosing turn will give you 2 mg. These are the ones that are available now. And the studies had to be done. So for example, if you look at this, the blue line is the original recombinant semaglutide or Ozempic, and then overlaid on that is the orange line. So again, a little bit of pharmacology. The peak that you reach is the Tmax. What time does it take to reach the peak? And that's identical to both. The peak that is sustained there is the Cmax. What is the concentration you achieve? Again, identical between the two. And then you have the area under the curve. What is the exact amount of the molecule that the patient is getting exposed to? Again, I would say almost identical between the two. Now, another word that we had to learn about when we were looking at this was something about bioequivalence and biosimilar. Biosimilar is only used to refer to a peptide if it is made by the recombinant DNA methodology. Then only it's called a biosimilar. If it is made purely by a synthetic manner, then it is called bioequivalent. There are some regulatory differences in the way these molecules have to be studied or tested. But basically, just to say that this is the difference between bioequivalence and biosimilarity, and some of the India is a big generic market, many bioequivalent drugs. Most recently, we got empagliflozin, DAPA, metformin, atorva, all of these are bioequivalent drugs. Biosimilars are the ones that are still made by recombinant DNA. So there you can see the monoclonal antibodies. Thank you. So again, another term that we learned about, bioequivalent and biosimilar. This was a clinical study using the Semalex molecule, the Torrent molecule, and the recombinant DNA Ozempic. And you can see that the HbA1c reduction is comparable between the two arms. This was at the end of 24 weeks. And weight reduction was also very similar between the two. No difference, saying that the synthetic molecule works as well as the recombinant DNA molecule. So to conclude, I took you on a journey where I talked about how initially peptides were removed from organs, which were cut up, grinded, acid, alcohol, all of that, to designer peptides, to the way the synthesis has also changed. Both the solid phase method. There is one more thing which is now apparently happening, which you'll be hearing about, that's called NCL, natural chemical ligation, which is supposed to be an even better step compared to this SPSS. Like I said, it's fascinating, the pharmacology. All of us, once we finish college, we don't think about it unless we study it. But I think the bottom line is whether it is produced by a synthetic method or recombinant biology, stringently purified peptide molecules yield identically safe and will give you the same efficacious clinical results. With that, I'll stop and happy for any discussion.